Hello friends and welcome to my channel Be Yourself. In my last video I have discussed the answer key for the part A of CSIR GRF Net Life Sciences. Now this time I'm going to discuss about the part B questions. So what I analyzed by I have gone through the previous year questions and this year questions, and what I have analyzed that the questions are not repeating, but the questions from the same topics are repeating. So why don't we prepare that topic which has been asked in this question, in this year question, so that we can have a better performance, or can we have a sure shot success in the upcoming examinations. So without wasting time, I'm proceeding with my first question, which is that consider the short double-stranded linear DNA molecule of 10 complete turns with 10.5 base pair per turn. The ends of the DNA molecules are sealed together to make a relaxed circle. This relaxed circle will have a linking number of, then we have to choose what would be the linking number. Now, from the questions, it is mentioned here it is 10 complete turns, and the linking number is how the first strand or the one strand is winding over the second strand. It means it has wind over the second strand by 3 times. Sorry, 2 times. One here and one here. Complete turn. Okay, 2 times. So we can say that the linking number for the particular strand will be 2. And here it is mentioned that 10 complete turns, so the linking number will be 10. Why don't we prepare for about more questions about the linking number in detail? Here I have two figures, figure 1 and figure 2. In this first figure, the linking number is 1, and in second figure we have linking number 6. Here you can easily visualize that the one strand and this second strand is crossed with one another only one time. But if you will see in this figure, there will be 6 places where the one strand is crossing over the other. So the linking number will be 6. This is only as in the case of relaxed state. And one thing is that when the coiling is left-handed, then the linking number will be negative. But when it is positive, right-handed, then the linking number will be positive. Now, there was mentioned that there was 10 complete turns, but what when there will the condition will not be mentioned, then there is a formula for linking number is that linking number for relaxed DNA is equals to base pair per base pair turn. Okay, this was in the case of relaxed state, but what if the DNA is supercoiled? If the DNA is supercoiled, then the linking number in supercoiled DNA is called a specific linking difference or the superhelical density, which is equals to the change in linking number by its original linking number. Then how to calculate this change in linking number? Suppose this is a relaxed DNA having the linking number of 100, then there will be two possibilities. That one is that it can either undergo positive supercoil or undergo negative supercoiling. If it undergoes positive supercoiling, suppose it has supercoiled by increment of 2, then the linking number will be plus 2. And when it has undergone negative supercoiling, then the change in linking number will be minus 2. And this only change in their linking number is termed as topoisomerase. These two DNA will be the topoisomerase. Now move to the second question. This is the signaling pathway where an ligand activin is binding with a receptor which is phosphorylated, and then it is giving rise to another molecule A which is also phosphorylated, and when it combined with B molecule it is transported to the nucleus and it is regulating the transcription. Before answering this question, I would like to explain you what actually this pathway is. This is a transforming growth factor beta signaling pathway. Okay, and this is a cellular process which is involved in adult as well as in developing embryo. In case of developing embryo, it controls cell differentiation, cell growth, as well as apoptosis. And apart from activin, there are some other ligands for the TGF beta. These are the bone morphogenetic proteins, also termed as BMPs, growth and differentiation factors, termed as GDFs, and anti-Mullerian hormones, or AMH. Activin, it was in the question. Other is nodal, and some are the members of other common members of TGF betas. Okay. Although they regulate the growth in the adult as well as developing embryo, but somehow they are different from each other. Look how they are different from each other. They are different in their receptor and function. For example, activin bind to the activin receptor 2, and it is specific for the embryogenesis and osteogenesis and regulate many hormones including pituitary, gonadal, and hypothalamic hormones as well as insulin. They also serve for the cell growth factor. Another is BMPs, and it has a receptor of 2, and its main function is osteogenesis, cell differentiation, anterior or posterior axis specification, growth, and homeostasis. Next is the TGF beta family, and it also has second types of receptor and involved in embryogenesis and cell differentiation, but apart them it also involved in apoptosis. The last one is nodal, and it has a common receptor of activin receptor 2b, 1b, and 1c, and it is responsible for mesoendodermal induction, patterning of nervous system, and determination of dorsal ventral axis in vertebrate embryos. So these are the different types of ligands which can also regulate the growth and differentiation function in the pathway. Now, I have discussed about type 1 and type 2 receptor. Then what is type 1 and type 2 receptor? This is type 1 receptor. I have represented it with the red color, and type 2 by the green color. This is the outside of the cell, cytoplasm, and nucleus, and where it is the chromatin or the DNA fragments where the transcription factor is there, and it will not lead to the synthesis of its messenger RNA until it is not regulated. So for its regulation, what happened? First of all, TFG's factors, beta factors, bind with its receptor, and as soon as it binds with its receptor or type 2 receptor, it will phosphorylate the type 1 receptor. After its phosphorylation, SMAD2/3 also get phosphorylated, and after that this SMAD2/3 then bind with SMAD4, and this SMAD4 assists its transportation towards the nucleus, and where it will regulate the transcription factor and lead to the formation of the desired proteins messenger RNA. Okay, these are the whole process which is get regulating. Here you can see that SMAD 2/3 is get phosphorylated, and this SMAD 4 is assisting its transportation from cytoplasm to the nucleus. So now again back to the questions. Here it is, you are also seeing some similar types of pathway. This would be SMAD2/3, and this B1 will be SMAD4. So the answer will be 4. No doubt in this. Okay, now next question. This is about the secondary antibody routinely used for the detection of primary antibodies in Western blotting experiments are anti-allotype, anti-idiotype, anti-isotypic, or anti-paratypic. Before answering this, we must know about the anti-allotype, isotype, idiotype, and paratype. So let's start. First, I start with the isotypic antibodies. These are the antibodies which are similar in their constant regions, or the light chains are almost constant, similar, but they vary only in their heavy chains. This is just like immunoglobulin M, which has the heavy chain of what we know it will be mu, and it will be G or the gamma. So You can relate this isotypic antibody as the five finger of an individual. It can be immunoglobulin 1, M, G, A, E, and D. Okay, it means same individual having different types of antibodies or the different size of your fingers. Now move to the second antibody, which is allotypic antibody. In this, if you will see, then the variable region is different in the in two. Although the immunoglobulin is same, immunoglobulin G1 and G1 is same, but in different strain of mouse A and B, this region will be different. Or you can say that the index finger of a male and index finger of a female. You can clearly analyze the difference between these two fingers or these two hands. This is suppose the immunoglobulin G1 of a strain A, and this is the of a strain B. Now move to the idiotypic antibody. Idiotypic antibody, if we will see in in the same individual, suppose we have taken the two strain of a single strain of mouse which is A, then a single antibody G1 will act differentially with different antigens. This you can see here the paratope, which binds with the epitope of the antigens, have the different binding site or different binding structure. This is termed as idiotypic antibody. You can relate this as you can apply different nail arts in your fingers. This is just like a idiotypic antibody. Now the question is that how we which types of antibody should be applied for the Western blotting. Now, if we will see with the isotypic, allotypic, and idiotypic, you will see that the specificity for binding towards an antigen increases from isotype, allotype to idiotype, because idiotypic is very specific to the antibody. Now, before answering this, I would also like to explain you about a very brief explanation about the Western blotting. Western blotting. What we do, this is for the analysis of the protein. Like Western blotting, there are some other blottings too, like Eastern blotting and Southern blotting and Northern blotting. I will explain this in all those in my upcoming videos, but in this video I am only explaining you with the Western blotting. What we do in this, we first disrupt the cell membrane to extract the cells or the content of the cell or cell organelles, and we are targeting here a specific protein. So what we do, we will apply for the differential centrifugation so that we can extract out the particular protein molecules. But we will not get the particular protein molecules. We will get the mixtures of the protein molecules. So what we will do, we will then pipette out the desired layer of our protein content, and we will undergo the SDS phase, which is the sodium dodecyl sulfate polyacrylamide gel electrophoresis. Then we will load our sample in the wells. Okay, we have here treated the protein with the SDS, which is sodium dodecyl sulfate. It has given a net negative charge to the protein. So when we will apply charges negative to positive, then the protein will start moving towards the positive charge. Now let's see, after applying voltage difference across this phase, what we will get. We will get a certain bands. The protein, the bands which has traveled far below will have the molecular mass less than that of the compared with the mass which has traveled less. This is because polyacrylamide gel electrophoresis will have a network of the mesh-like structure, and the small particle can only escape from such meshes and can travel further as compared to the larger molecules. Now, what we then do, this is we are having the top shoe of that gel. We will then place a nitrocellulose membrane over the gel. Then again we will apply charge. Now this time we have applied the charge from the gel towards the membrane. Then what will happen? That the the bands will be transferred to the nitrocellulose membrane. After that, we will remove out the nitrocellulose membrane, and we will have something like this, like views. Suppose uh, this is the desired protein, okay, or the bands of the protein. The next step is how to know whether this is really the desired band of the protein or not. Then what is done? Then we will apply the probing, or the probe means to identify that particular bands which we need. So for that, what is done? Antibodies are applied specific for that protein. Okay, but these antibodies, you can look here, you can have seen here, it have bind to this particular band. Now this, how we will analyze it where it has bound, because there is no such any reporter bonded to the antibody which can tell that here is the band which you are looking for. So then what the next step will be? We will wash out the membrane so that the unbounded antibody will be washed out, and then we will apply again the antibody which have a certain probe. It can be any fluorescence compound, it can have be any enzyme. If you remember the ELISA, where there we were using certain enzyme, and the substrate when react with in presence of certain enzyme, it have given some color to the solutions. So similarly, like that, we have applied here and tagged a molecule which can be a fluorescent compound or any enzyme or any substrate which can give a color compound for a particular band, and we can easily analyze that band. Now again we will wash out the unbound antibody, and then what we will see, if the tag substance is any color any chromogenic, then it will give the color to the band, and if it will be the fluorescence compound, then it will give the fluorescence compound. Now from here we can analyze that this band we were looking for, or this was the protein or the desired protein. Now the question, again back to the questions. uh, Anti-allotype, anti-deutype, and anti-isotype. We are looking for a particular type of protein which is very specific. And if we will again look about the specificity, then it is increasing from isotype to idiotype. So what type of antibody we should use the idiotypic type. That another was the paratypic. Paratypic is the site where the antigen is bonding. But if you have looked the Western blotting, then if it is the primary antibody, the secondary antibody is binding with the Fc region, then there is no use of this paratypic region. So we will fourth option cannot be there, and we are looking for the specificity. So the specificity is the idiotypic. So here the answer will be anti-idiotypic. So the option will be the third one. Sorry. So the answer will be anti-idiotypic. Now move to the next question. That is, which of the following event will not usually lead to the formation of normal cell into cancerous cells? First option is the gain of function of oncogenes. It's obviously if the normal gene is converting into the oncogenes, it will develop into cancer. Loss of function of tumor suppressors. If tumor will not be suppressed, then it will lead to the cancer. Third is gain of function of genes involved in nucleotide excision repair. This will be the answer. Why I am telling you? But once also look at the fourth number, which is the loss of function of pro-apoptotic related genes. Pro-apoptosis means the genes which will initiate the apoptosis process. If the apoptosis will be there, there will be no chances of the survival of the cell. So the answer will be 3. And what is the nucleotide excision repair? It is a repair mechanism. If your DNA is damaged, then it will lead to the cancerous or some other mutations. But if it get repaired, then the cell will be the normal one. So what is the nucleotide excision repair? DNA lessons that cause large distortion in the helical structure of DNA generally are repaired by nucleotide excision system, a repair pathway critical to the survival of free living organisms. Okay, next is that in this repair system, a multi-subunit enzyme hydrolyzes in two phosphodiester bonds, one on either side of the lessons or the where the DNA is damaged. In case of E. coli and other prokaryotes, enzyme system hydrolyzes the fifth phosphodiester bond on the 3' side and 8th phosphodiester bond on the 5' side to generate a fragment of 12 to 13 nucleotides. And in the case of human and other eukaryotes, it will cut down at the 22nd phosphodiester bond toward the 5' end and the 6th phosphodiester bond toward the 3' end, and so it will generate 27 to 29 nucleotides. Okay, so let's have a look for this. It actually requires the assistance of certain proteins, which is UVRA, 2 unit of UVRA, 1 unit of UVRB, and 1 unit of UVRC, and this all together is known as ABC-XC nuclease. What it do? First of all, UVRA and B will scan out the DNA and find out where the lesion is. When it is analyzed, then UVA will be spliced out and UVRC will bind to the UVRB, and it will then lead to the excision of the base pairs at the 8th side of the 5' and 5 base pair toward the 3' end. Then it will be these DNA lessons will be cut out, and then DNA polymerase 1 will act over this strand for the synthesis of DNA or the spliced out DNA. Then it will now we have the DNA which is the repaired one DNA, or the DNA having the complementary sequence with that of this yellow strand. Okay. So, and now you can see the gap was there, then this gap was healed by with the help of DNA ligase. Now we have the repaired DNA or the DNA without any damage. So you can see here, if the DNA is repaired, then the cell will be a normal cell. It cannot be a cancerous cell. So the answer will be the third option. In case of eukaryotes, same process will continue as this was example for the prokaryotes. Only the difference was that instead of polymerase 1, which also used for the nick translation, DNA polymerase epsilon will be used in the case of eukaryotes. Next question is that which one of the following is the foodborne toxins? Without any doubt, it will be botulinum toxins, as it causes the food poisoning. Even the canned food can grow in the canned food because it is bacteria which has the spore forming activity, and we know that it is very difficult to kill the spore forming activity bacteria. It can tolerate even harsher to harsher condition. Even if we will autoclave it, it will not die. Even we will apply a large amount of heat or high heat, even then it will not die, because it has changed to the spore and the unfavorable condition. So the best way to kill such type of bacteria is that first sterilization, then again back to the incubation process where the spores can get the desired temperature for its germination, and when it gets germination, then again go for the sterilization process. So to kill such types of bacteria, it is better advised to repeat the process of, or the reverse or alternate the process of sterilization with that of the incubation. I'm stopping here now, because if I will continue some more questions, you will become sleepy. So I have discussed today only 5 questions. In my next video, I will discuss some more questions of part B. So I'm sorry for your, I'm also thankful for your patience to wait for my next videos. I will soon update you with them with more videos. Till then, goodbye, and don't forget to subscribe and share my channels to when, so that when I get update you with the new the new questions, you can update it as soon as possible. Till then, goodbye and all the best for your future examinations, and be yourself.